Hello, my name is Twumasi-Ankrah, and I will be recording an informational video on the cell cycle and how it is regulated, as well as consequences when certain regulations are not regarded. To begin, the cell cycle is a series of events that takes place in a cell, leading to its replication and division that produces two identical daughter cells. In prokaryotes, the cell cycle occurs via binary fission, and in eukaryotes it is used to create somatic or body cells. As seen in the picture shown on the screen, the cell is going through different phases of this cell cycle. For example, this right here is this is known as interphase. This is where the cell spends about 90% of its life. It just grows and synthesizes its DNA, replicates it. Usually there is only one cell at the beginning of this phase. But I didn't have a better diagram. So just imagine there's one to begin with, and the cell is as I said, growing, and then during prophase, chromosomes condense from chromatin to chromatid, so the strands become shorter and thicker. They're more visible. Prometaphase, the nuclear membrane begins to disappear, spindle fibers begin to form, and you can see the centrioles moving. They start to move. Centrioles then connect spindle fibers to the kinetic cores of the sister chromatids during metaphase, where they line up on the metaphase plate. And during anaphase, they begin to divide. And in telophase and cytokinesis, the nucleus has split, and in cytokinesis, the cytoplasm, as in the rest of the organelles, have also split. And you can see in the end, the result is two diploid daughter cells that are completely identical. Now, control of the cell cycle is necessary for a couple of reasons. First, if the cell was not regulated, cells would constantly undergo division. And while this may be beneficial to certain species, constant division without cause would be biologically wasteful. Secondly, internal regulation of the cell cycle is necessary to signal passage from one phase to the next at appropriate times. This regulation is not achieved through strict time constraints, but it's rather achieved through feedback from the cell. And to show the process where the cell is during its life, this simple diagram will be used. As stated before, interphase is where most of the cell spends its lifetime. About 90% of its life is spent here, growing and replicating DNA. Replication of DNA only happens once. Interphase is broken into three phases: G1, the growth phase, S, synthesis, and G2, growth 2 phase. During this, and during G1, these feedback reports are conducted at 3 checkpoints, obviously, during 3 stages of the cell cycle. The first checkpoint, G1, occurs during the G1 phase of interphase, hence the name. It's also referred to as restriction or start checkpoint. And during G1, the cell is metabolically active and continuously grows with a sufficient amount of nutrients and other necessities. So it creates proteins, it makes sure the cell is big enough, because that is important to continue on throughout the cycle, to make sure that cells are completely identical. About 2/3 of the way through this phase, the first checkpoint is reached, the restriction checkpoint, and here the cell assesses several different factors. First, it needs to assess the size of the cell. The cell needs to be big enough so that it can hold the right amount of DNA, it can split properly, it can be the right size, basically. It needs the amount of proteins, so the cell creates several different proteins to get the size of the cell up there. The environment also needs to be optimal so that the cell is not running into several unnecessary hindrances. There needs to be enough room for the cell to grow, obviously. The nutrient level of the cell needs to be high enough, so the nutrients are checked. The cell makes sure that it's getting the right amount of nutrients so that it can sustain the cell throughout its entire lifespan. And the growth factor presence, it helps, it provokes the growth of cells, basically. It speeds that up. So the cell checks to see if there's enough of those there. And if these, along with several other factors, are deemed adequate in the cell moves on to the synthesis phase, which is the next phase of interphase. The DNA replicates, and the checkpoint, the DNA replicates, and the cell moves on to the next phase of interphase. I'm sorry, it extends all the way over here. The interphase extends all the way over there. But it moves on to the next phase of the interphase, which is G2, the growth 2 phase. And the cell replicates organelles in preparation for mitosis here. This is also the last chance the cell has to make more protein to grow before division, which is mitosis. And at the end of this phase, the second checkpoint is reached. And the checkpoint checks to see if DNA has been replicated correctly from synthesis, because if DNA is not replicated correctly, then the daughter cells will not get the right amount of DNA chromosomes that they need. There also needs to be a sufficient amount of nutrients. And as for the same reason as before. And the environment needs to be favorable once again. And if the cell checks out of these checkpoints, then it is moved on to the mitosis stage of division. The last checkpoint, M, occurs right about here. It stands for metaphase. So this is mitosis. This is the nuclear division. It's broken into prophase, metaphase, anaphase, telophase, and cytokinesis. And during metaphase, the spindle fibers condense, the chromosomes align at the metaphase plate, getting ready to split, and the last checkpoint checks to see if the chromosomes are aligned at the spindle, as seen in the picture, the slide before. The chromosomes need to be aligned at the spindle correctly. Homologous chromosomes cannot be paired. Proper pairings need to be checked, and it checks to see if the cell is ready to split. And if the cell passes, it completes the cell cycle, and 2 new daughter cells are formed. And if any of these checkpoints are not passed, the division is delayed until issues are fixed. But if issues cannot be fixed, the cell moves into a state outside of the cell cycle called G0. The cell does not divide here; it's a resting state. So it is removed from the cell cycle, essentially. Next, we will be discussing more specifically the proteins that interact to regulate the cell cycle. The checkpoints that we described earlier were established by proteins that use cues from the cell's environment to trigger entry and exit from distinct phases of the cell cycle. And 2 main families will be discussed: CDKs and cyclins. CDKs are cyclin-dependent kinases, which are enzymes that phosphorylate proteins to send a signal that the cell is ready to pass into the next stage of the cell cycle. The cell will not progress if not enough CDKs are made. So CDKs are activated when they are bound to cyclins, and cyclins are another family of proteins that undergo constant fluctuation throughout the cell. So their numbers are always depleting and increasing at several different intervals, and it's created and degraded repeatedly, so the availability in the cell varies. When synthesized, they act as an activating protein and bind to CDKs to form a cyclin-CDK complex, which signals a pass into the cell cycle phase. The cyclin degrades once again, and it deactivates the CDK, which signals an exit into a particular phase. Now, in this picture, these are known as the CDKs. These are the cyclin-dependent kinases. And cyclins are right here. I won't label them as just C from now on. Cyclins, these are cyclins. And during G1, you can see that there are no cyclins here because they're deemed like they're not necessary in G1. So there are no cyclins present here. So, but during the S phase, you can see that there's a buildup of cyclins. So the cyclin amount accumulates. Cyclins accumulate here during S. And during G2, you can see that cyclins and CDKs have bound to form this CDK cyclin complex. And after mitosis, once again, the cyclin is degraded. And this is because mitosis has already occurred, the division of the cell has already occurred, and the cell is moving back into G1, where we earlier discussed that cyclins are not necessary, so cyclins will be degraded. You can see this accounts for the fluctuations of cyclin availability. So basically, synthesis of cyclin begins in late S phase. It continues throughout G2, and because it's forming in this stage, it accumulates, and when it accumulates, it combines with many CDK molecules, which allow it to send signals throughout the cell that the cell is ready to pass into mitosis, which helps divide the cell. And during anaphase, when the component of the MPF is completely degraded, the CDK is deactivated because cyclins are degraded, and the cell moves back into G1. This is an example of a mycotic cyclin, which would be MPF. The cyclin accumulates gradually during G2, as seen here. At once a high enough concentration, they can bind to CDKs. When this complex forms, it acts as a signal for the G2 cell to enter mitosis. And once the cyclin degrades, the MPF is inactivated, and the cell exits mitosis by dividing into two cells, as we saw in the first slide of this PowerPoint. And it re-enters G1. See, the cell cycle continues over and over again. It's a cyclic process, obviously. The cellular signals described earlier, the cell size, DNA replication, completion, environment, they provide signals that regulate the synthesis and degradation of cyclins. Now, this is just a graph to show process of cyclins. This is the M phase. As you can see, wait, let me just label this quickly. G1S, G2. It repeats. Now, you can see that, I'm sorry, it's my, okay. There's a low, sorry, I have to label this cyclin concentration and MPF activity. Now, it is seen that the cyclins are, at mitosis, there's obviously a high concentration of cyclins. There is a buildup of cyclins from S to mitosis, and the MPF activity is quickly rising. And this accounts for the fact that cyclins help to divide the cell. So they're needed in mitosis, obviously, which is why they're produced. And after they're used, cyclin concentration once again fluctuates, it depletes, and you can see that there's not much until late S phase once again, as we stated earlier. Now, lastly, the loss of the control of the cell cycle is one of the critical steps in the development of several diseases such as cancer. And in cancer cells, the normal systems that prevent cell growth and invasion of other tissues are disabled, so these cells grow in the presence of signals that would normally inhibit cell growth, but instead, these cells rapidly grow, they invade other cells, it could potentially kill the organism. So, cancer cells do not heed to any regulations, and which is why the field of study is being actively researched to find a cure, because without these regulations, how is the cell supposed to stop growing? It doesn't have regard for density dependent. It doesn't have regard for internal or external factors that inhibit cell growth, that regulate cell growth. So this is basically it for this PowerPoint. Thank you for joining me today, Simon. Have a nice day. Thank you.